collaboration in mental health research between the UK and India. And since then, I've been really looking for an excuse. I was so blown away, partly by the science, but also just by this fantastic city, that I was always looking for an excuse to come back and collaborate. So we were really delighted last year to get funding again from the MRC to establish pump priming awards to set up collaborations, not just scientifically, but also in the context of training between the UK and India. That's really what's funded this initiative, and I really hope that not only in the context of training, but also in the context of the really exciting data we're going to generate together as part of the project, that we will collaborate for many years to come. So thank you very much. Okay, I will. So for those that don't know, so the real focus of the work in our group is on understanding both causes and consequences of molecular variation in the brain, and we're interested in understanding that in the context of a whole range of neurodevelopmental, neuropsychiatric, and also neurodegenerative diseases. And in the kind of context of this talk, I'm going to give you initially an introduction to why we think epigenetic variation, epigenetic processes are important in mediating the onset of these phenotypes. And I'll give you some kind of snippets of some of the work that we're doing, and Emma and Joanna will give you a bit more detail about specific aspects of those studies in their talks later. And I want this to be really informal, so if anyone has questions as I go through, because I think there's a fairly long slot, please interject if I'm not clear about anything. Please feel free to ask a question, and I'll do my best to answer. So one thing that everyone has asked me since I've been in India is, where is Exeter? So I thought I should start really with a map. So Exeter is a small city, certainly compared to Bangalore. It's located in the southwest of the UK in a county called Devon, and you can see where that is there. And it's a really beautiful area of the UK. So one of the things that appealed to us was the scenery around there, and it's a really great place to live and grow up a family. And so this is the kind of postcard picture of Devon that is often touted by the tourism board. Unfortunately, the reality is often very, very different. So this is a normal scene in Devon, and so it's obviously often quite miserable in terms of the weather. So again, it's a real pleasure to be here and have some sunshine and some warmth, especially at this time of the year, where it's a particularly dour place to be in the UK, cold and wet and rainy. And I also want to just put up this figure at the start, because this is a picture of our group. These are the people that do all the work, and you can see Emma and Joanna, but also the people that fund the work that we do. And really, this is we work across the spectrum of CNS diseases and disorders, and we're funded by many charities and funders in this field, and we're obviously really grateful for that. The other thing is we have a lab website, so if anyone's interested in learning more about what we do, you can look on there, and for anyone who uses social media, you can follow our updates on Twitter, and we post papers and things on there. Okay, so this is really known to everyone, but I always like to start by putting the problem in context, and this is data from the World Health Organization that is outlining really the global burden of non-communicable diseases, and you can see the kind of huge proportion, almost a third of this taken up by neuropsychiatric disorders, and that in this context includes schizophrenia, a range of other neurodevelopmental disorders, but also Alzheimer's disease and other types of dementia. And this often surprises people when I talk to non-psychiatry audiences, particularly in the UK, where there's a lot of emphasis really on cardiovascular disease and on cancer. But unlike kind of these other types of disease and disorder, we actually know relatively little about the etiology of mental health. Certainly, as you all know, there are no definitive diagnostic tools, so you can't go to your GP or doctor and get a blood test that will tell you that there's a problem, just like you would for measuring blood pressure or other types of more peripheral disorder. As we all know, there are no cure for these diseases, and the medications that are used to treat them are often fairly ineffective and obviously have many side effects. And there's a real kind of hole at the moment in terms of pharmaceutical development of novel therapeutics. And one of the kind of applications of the type of genomic data that we're trying to generate is to develop new targets that can be used in the development of novel drugs. And the other context I want to put my talk into is in the context of the real advances in terms of understanding the genetic etiology of mental health. So again, through a real kind of advert for collaborative science and the real successes that can be derived from people working together, I think the genetic understanding of schizophrenia is a real prime example of that. So the Psychiatric Genomics Consortium has now put together large numbers of samples, and obviously the technology now exists to characterize genetic variation across the genome at relatively cheap prices. And we've gone very rapidly from a stage maybe 20 years ago where genome-wide association studies were done at what at the time was seen like very large numbers, so, over 3,000 samples, where we identified 0 genome-wide significant loci associated with schizophrenia. And at that time, I think there was real depression in the field because people thought this was going to be a really insurmountable problem. But by putting together large samples, we're now at a stage where we have really actually in the latest World Congress of Psychiatric Genetics in Glasgow last year, sample sizes of over 70,000 individuals with over 250 regions of the genome known to harbour robust genetic variants associated with the disease. So this is really fantastic, I think. But it leads to a new challenge, and that's really, what do we do next? How do we improve understanding of the actual biological effects of these genetic risk variants? And as I'll talk about later, that's one of the kind of key focuses of our work. So a lot of the regions of the genome that are shown to harbour these risk variants for schizophrenia are very large. They maybe encompass tens of genes, and actually working out what is the causal variant and what is the mechanism by which that variant leads to some neurobiological change that ultimately leads to disease is still very much not really understood. So I think this cartoon really kind of sums up the kind of current situation. So we are really getting to grips with understanding that the genetic, and I've put polygenetic risk factors associated with disease. Epidemiological studies are telling us a lot about the environment. We know things like stress and lifestyle and other exposures are fairly robustly associated with diseases like schizophrenia. But how these things come together to change the way that genes are expressed, maybe in a specific cell type or at a specific point in development, a specific region of the brain, is still very much misunderstood. This is really what our work is attempting to focus on. And I also want to put this in a bit of historical context, because actually the concept of epigenetics in schizophrenia in particular is a relatively new concept. And actually the first mention of it in the literature is from 2 eminent psychiatrists who are working at the Institute of Psychiatry in London, Irv Gottesman and James Shield. And in 1982, they published this book called the Schizophrenia, the Epigenetic Puzzle. And in many ways, this was ahead of its time in how they thought and conceptualized the origins of schizophrenia. And so I've just put up a couple of quotes from this book. So here you can see it says that the genetic pieces provide more of a figure. The environmental ones provide the ground, sky, and the other scenery. To repeat a truism, genes and environment are both essential for the development of any human characteristic, and the puzzle is epigenetic. The task ahead is to discover how genes and environment interact to produce schizophrenia. And I think that still really remains the question that we're trying to tackle today. And the other thing they say, which is really true for anyone trying to undertake this work, and again, I think they really tap the essence of some of the problems of this work. It says, of course, it is much easier to talk about such epigenetic interactions than to design experiments that will elucidate them. So I hope throughout this talk and throughout some of the workshop sessions that we'll put on tomorrow, that you'll get a feeling of actually not only the importance of some of these processes, the importance some of these processes may play in disease, but also the real difficulties there are in actually teasing out those processes and some of the experimental limitations that make finding real causal links between epigenetic variation and disease very difficult. And this is, again, a key figure from that book. And again, I think this kind of highlights some of the things that we're really still only starting to understand. So this is really just looking at how genetic burden itself is not necessarily the only thing that leads to the development of a disease like schizophrenia. And in fact, how that if you have two identical twins who share the same genetic burden, they can often end up with very different phenotypic outcomes. And one of the focuses of our work is very much on looking at identical twins and working out why they may be different in terms of a disease like schizophrenia. But also that individuals who have a low genetic burden may still go on to develop a disease like schizophrenia. And of course, in individuals with a high genetic burden, we're still trying to understand the actual molecular mechanisms by which those genetic variants lead to disease. And these are all aspects of the work that we are trying to do. So one thing that's becoming increasingly clear is that for the whole spectrum of complex disease phenotypes, there are many epidemiological features that tell us that there is likely to be much more than DNA sequence variation involved. And I've put a few of these up here. So as I mentioned, there is often discordance between genetically identical monozygotic twins, not only for the prevalence of the disorder, but also in terms of maybe how that disorder manifests in terms of clinical symptoms, age of onset. Many of the disorders that we're interested in studying have big gender differences. So we do a lot of work on autism, for instance, where prevalence rates are much higher in males than females. Conversely, prevalence rates for depression are higher in females than males. Many of these traits and phenotypes have a very fluctuating disease course. And we certainly see kind of over the course of a lifetime, there are peaks of disease following key points in development or following kind of key hormonal changes in an individual. And of course, we know that the environment plays a really important role, but there appear to be very narrow windows of sensitivity, periods during which an environmental exposure can be considered a really important risk factor. But then, of course, during other parts of the life, those same environmental influences may not infer such a risk. And of course, we see non-familial sporadic cases, so there is clearly a genetic influence, but these do not follow clear Mendelian inheritance patterns. And all these things together tell us that things other than DNA sequence variation is likely to be important. So this is how geneticists often think of the genome, this sequence of A's, C's, G's and T's, and of course what genetic association studies are doing are looking for differences in the frequency of variants in this genetic code in individuals with a disease compared to groups of individuals who don't have a disease. And of course we can now sequence whole genomes very efficiently, very cheaply, and these have told us a lot about how this genetic variation is actually obviously the key driver of evolution. So we now know and can map the genetic differences that distinguish us from our closest evolutionary neighbours. But if you take a brain from a human and a brain from a chimpanzee, they're both clearly a brain. There's slight size differences, a few functional and structural differences, but these are both clearly brains. But if you take the audience here, each of us is made up of many hundreds of different cell types. Each of those cell types has exactly the same genetic information contained within it. But of course, if you look at them under a microscope or you look at their functional importance, you'll see that they do very different things. They look very different, and that's driven by the fact that each of these different cell types expresses a very different subset of genes at different levels and at different key points in development. And of course, the brain is a key example of that. So we know there's huge amount of neural diversity in the brain. And some of the kind of new methods that we're now embracing and approaching that have been driven by the kind of genomics revolution, single cell RNA sequencing studies, have really started to highlight the huge amount of diversity in cell types in the human central nervous system, each characterized by a very specific pattern of gene expression that defines the actual physiological phenotype of those different cells. And you only have to really look at the way that our genetic material is stored to realize that it's much more complex than that linear sequence of A's, C's, G's and T's. So this is a really beautiful image of a human mitotic chromosome. And you can see the very ornate way in which our genetic material is stored and packaged. So the DNA material is coiled around these core histone proteins to form chromatin. And of course, that's all condensed in together to form the actual chromosomal structure that you see in the image behind. And it's this structure of how the DNA is packaged and stored that really mediates how accessible the actual DNA sequence is to the factors, the transcription factors in the cell that bind to that DNA and actually drive gene expression. And in many senses, when we talk about epigenetic processes, this is really essentially what we're really thinking about and trying to understand. And just to put this in a bit of historical concept, so the term epigenetics was really first used in kind of popular terms by a British biologist, Conrad Waddington. And he used this very nice analogy of an epigenetic landscape, a marble rolling through this epigenetic landscape to describe how cell fates are established and maintained during development. So a cell going from its kind of initial totipotent state where it can essentially derive into all the cell types that make up an organism, becoming more and more differentiated and going down these different valleys until it's at a point where it's unipotent and totally differentiated. And there are kind of many ways of describing this analogy. So this is Clapham Junction railway station. This is in South London, if any of you have been there. It's the busiest railway junction in Europe. I'm sure there are much busier railway junctions here in India. But you could imagine, really, it's how important it is for a train to, the signals, the train to go in the right direction, to make sure it gets at the right stage. And really, this is, I think, a nice analogy for how the different differentiation of cell types across development. So if the signals are wrong or a train ends up going down the wrong track, then there will be all sorts of issues and problems. And another kind of very generalizable analogy, you can think of your DNA sequence really as a piece of sheet music. your phenotype, the way a cell expresses its its genes as maybe an orchestral performance, and your epigenotype or your epigenome really is the interpretation of that piece of sheet music towards the actual orchestral performance. And you can see all the marks and things that the conductor has written on there to get that interpretation correct. And you can think about epigenetic marks, modifications in the same type of way. So why is this important? Well, you can imagine two identical stretches of DNA with different epigenetic scenarios. So on the left, the epigenetic status means that there's lots and lots of gene expression, and that leads to lots and lots of protein being made. And on the right, a very different epigenetic profile that leads to very limited gene expression and no protein being made. And what this tells us is that the DNA sequence determines the actual molecule, the actual mRNA molecule that's synthesized, but the epigenetic regulation or the epigenetic marks that define the expression determines how much and when and where that specific mRNA molecule is made. And the follow-on from that is that even genes that don't harbour or contain a variant or polymorphism that may be associated with disease can still be pathogenic if they're expressed at the wrong time or the wrong amount or in the wrong cell type. So it provides a way by which genetic regulation can still lead to phenotypic variation independent of the DNA sequence. So there are many different types of epigenetic modification, and the one that most focus has been on, particularly in the context of psychiatry, is on DNA methylation. And this is simply the addition of a I don't know if this works. I don't think it does, but the addition of a methyl group. You can see the CH3 here to the fifth carbon position of cytosine. That's one of our the four base pairs that make up our DNA sequence. And the very kind of crude consequences of this is, firstly, it acts to displace certain transcription factors, but it also attracts proteins that are attracted to methylated DNA. And again, these have some of those structural conformational effects on the genome that I described earlier. And in reality, the situation is much more complex than that, and I'll touch on that later. Another modification to DNA that we are becoming particularly interested in is DNA hydroxymethylation. This is a relatively recently discovered modification to DNA that was initially assumed to be relatively transient as part of the actual demethylation pathway. It was assumed to be present at fairly low levels across the genome, but that actually turns out that it's specifically enriched in the brain, particularly in certain neuronal subclasses where it's actually fairly stable. And we've recently described the patterns of this modification across different regions of the human brain, and shown how it's likely to play just as an important role in gene regulation as DNA methylation. And in reality, there's a whole kind of suite of epigenetic modifications that not so much act to switch on or off genes, as you may kind of hear in a textbook. In reality, they more act to fine-tune genomic function across development. And we and others, via projects like the Epigenomics Roadmap Initiative, all part of the International Human Epigenomics Consortium, have set out with the aim of mapping these whole layer of epigenetic modifications, both histone and DNA modifications and other types of structural information across the genome in all the different cells and tissue types that make up an individual. So obviously mapping the human, sequencing the whole human genome was a gargantuan task at the time. But actually the next stage of this process, actually mapping the different layers of gene regulation across the genome in each of the different cells and tissue types, is an even much more complex task that's only really starting to happen in earnest, driven by advances in sequencing technology. So this is a figure from one of the kind of key papers of the first set of output from the NIH Epigenomics Roadmap Consortium. It's just an example of a stretch of DNA, and you can see all the different modifications there that are layered across that stretch of DNA. And here, the kind of key message is that different cell types are characterized by very distinct epigenomes that mirror the developmental relationships between cell types, tissues, and lineages. And actually, a common developmental origin is the primary determinant of global epigenetic patterns. And in reality, if we really want to understand the role of epigenetic dysfunction in health and disease, this is really a key first step. It's actually understanding the normal patterns of epigenetic regulation across the cells and tissue types that we might be interested in the context of that disease. So in our case, that's obviously specific regions of the brain and specific cell types within those regions of the brain. One of the limitations of these early initiatives, ENCODE and the roadmap,